Hello everyone. Today I am delighted to share with you recent progress from an ongoing project where the goal is to better understand the functional role of individual gut microbes in celiac disease. More specifically, I will be talking about how we can functionally profile the gut microbiota of celiac patients at the species level resolution by using metagenome derived genome-scale models of metabolism. Celiac disease is an autoimmune digestive disease which occurs in genetically predisposed individuals in response to the ingestion of gluten, which is a protein found in wheat, rye, and barley. In fact, celiac disease is the only autoimmune disease for which both the genetic risk factors and the environmental trigger are well known. Nevertheless, research shows that genetic makeup and gluten exposure are necessary but not sufficient to trigger the development of the disease. And this supports the notion that this is a multifactorial disease and other factors may also be involved in its etiology. For example, researchers have previously looked into environmental risk factors other than gluten exposure, such as C-section delivery, formula feeding, antibiotic usage, and so on. An additional factor which has recently emerged as a key player in celiac disease etiology is the gut microbiota. However, despite the relatively well characterized associations between the gut microbiota alterations and celiac disease, we still know very little about the biochemical pathways and molecular mechanisms through which the gut microbiota interacts with host and environment in celiac disease. And more importantly, we still know very little about how individual gut microbes contribute to the disruption of the gut homeostasis and the loss of gluten tolerance in celiac disease. And this is for the most part due to a number of technological limitations which do not allow us to directly gain any insights into inner workings of the gut microbiota. For example, by using metagenomics, which is the state-of-the-art technology for microbiome profiling, we are able to determine the microbes that are present in a sample and their relative abundances, or to determine the functional pathways that are encoded by the metagenome and their relative abundances. Likewise, by using metabolomics, we can determine the metabolites that are present in the sample and their abundances. However, none of these technologies could provide any direct information about what microbes carry what pathways, or what microbes produce what metabolites, and to what extent, and what other microbes may possibly consume these metabolites. Addressing these questions is really key for understanding the role of the gut microbiota in disease, and more importantly for designing therapeutics. For the remainder of my talk, I would like to hopefully convince you that we can address some of these knowledge gaps by using computational genome-scale models of metabolism. A genome-scale model, or GEM for short of metabolism, is an exhaustive list of all metabolic reactions encoded by the genome of the organism. These models are constructed from the annotated genome of any organism and can be computationally simulated under a defined growth medium by using a class of mathematical modeling approaches called constraint-based analysis methods, which are based on a constrained mathematical optimization problem, to computationally predict the growth of the cell in that defined growth medium, and to predict the fluxes of intracellular metabolic reactions, and more importantly to predict what metabolites are possibly excreted out by the cell in that defined growth medium and their secretion levels. These constraint-based methods were initially used to model individual organisms or monocultures, but they were extended over the past decade or so by us and by others to additionally model interactions among different cell types, such as interactions among different microbes in the microbial community. So we provide here as the input the gems of metabolism, for example, for microbial species that are present in a community, and then for a defined growth medium, which is determined by diet for human-associated microbiomes, we can use these community-level constant-based methods to computationally predict the growth of different microbes in the community, and more importantly to predict what metabolites are possibly exchanged among the community members in the exchange levels. Therefore, this approach is able to address the knowledge gap that I talked about before, that is, which microbes produce or consume which metabolites in the community. So we used this modeling paradigm to study the gut microbiota of celiac patients by using fecal metagenomic data from a prospective longitudinal birth cohort study of celiac disease called the CDGEM study. So this study recruits infants less than 6 months of age with a first degree relative with celiac disease, and follows these infants until 10 years of age. So during the course of the study, some of these infants develop celiac disease and some of them they do not. And a lot of biospecimens such as fecal samples and blood samples among others, and a lot of metadata are collected from these subjects longitudinally at multiple time points. So in fact, we had previously analyzed the fecal metagenomic and metabolomic data from this cohort study by using microbiome-wide association studies and statistical methods, which you can read more about the details of these studies in our recent publications. But basically what we got out of these microbiome-wide association studies were a list of microbial species or strains, functional pathways, or metabolites that were differentially abundant between subjects who developed the disease and those who did not, or between different time points. And in these studies, we were not able to directly link these three microbiome features, and the farthest we could go was just performing a correlation analysis between microbes, functional pathways, and metabolites. And today I will be talking about how we can mechanistically link these three features by using gems of metabolism. So for our studies, we focused on 10 subjects who developed celiac disease or cases, and 10 matched controls at the time of disease onset. This is the work of two very talented students in my lab, Izzy and Anelia. We start with metagenomic data for each fecal sample, and then we perform taxonomic profiling to determine what microbial species or strains are present in each sample. And then we extract or construct metabolic models for each species in each sample by first mapping these species into databases of previously reconstructed gems of metabolism, such as the Agora resource from Ines Thiele's lab. And if they could not be mapped to an existing gem of metabolism, then we map them to databases of isolate genomes or databases of metagenomically assembled genomes or MAGs, and then we can construct new metabolic models from isolate genomes or MAGs. So this way we get the gem of metabolism for each species that is present in each fecal sample. So the next step then would be to integrate these individual species models into a community model, and we do this by following the same procedure as in the microbiome modeling toolbox, again from Ines Thiele's lab. We implemented everything in Python with some minor modifications, but briefly what we do here is that for each fecal sample we integrate gems of individual species present in that sample into a community model which has 3 compartments. In particular, microbial species are located in the intestinal lumen, and we also have a diet compartment and a fecal compartment. We additionally incorporate species abundance data from taxonomic profiling into this community model by constructing a community biomass reaction which represents the biomass production for the entire community, where the reactants of this community biomass reaction are the biomasses of individual species present in that sample, and the stoichiometric coefficients are the relative abundances of a species in that sample. We then computationally simulate this the community model by using community level constraint-based methods under a specified diet. So for our simulations, we simulated a typical Western diet by using an American average diet whose composition was available, and then by solving the model we could predict the growth of different microbes in the community, and we could also predict which microbes produce what metabolites or consume which metabolites in the community for that diet. And furthermore, we can predict the metabolites that are secreted into feces by the entire community. So we performed one such simulation for each metagenomic sample, and then for the first part of our analysis we compared the metabolites secreted into feces in cases compared to controls. For the brevity of presentation, here we have shown only the top 25 metabolites with the highest fold change in the production level in cases compared to controls. Some of the metabolites that were produced at higher levels in cases compared to controls were previously implicated in celiac or other autoimmune or inflammatory conditions, whereas some of the metabolites that were depleted in cases and are produced at higher levels in controls were reported to have anti-inflammatory or protective effects. So if you do not have metabolomics data for your samples, Gems of Metabolism will allow you to gain insights into how the gut metabolome looks like for your samples. But if you do have such data, you can get similar information from metabolomics as well. However, by using Gems of Metabolism, we can go even one step further and link microbial species to metabolites that might be implicated in disease pathogenesis. So what we did here was that we extracted from our simulations all species metabolite linkages representing whether a microbial species produces a specific metabolite. We uncovered close to 1000 such species metabolite pairs, out of which 45 turned out to be significantly different between cases and controls. In this heat map, we have shown the top 20 such species metabolite pairs with the highest fold change between cases and controls. Looking at one example, we have Predixal, a metabolite which also showed up in the graph that I showed in the previous slide. This metabolite is produced in our simulations at higher levels in cases compared to controls, and additionally the higher levels of this metabolite has been previously implicated in celiac disease. So looking at this heat map, we can see that this metabolite is produced by Rominococcus lactaris, and interestingly, higher abundances of this species has been previously implicated in other autoimmune and inflammatory conditions. So by using Gems of Metabolism, we are able to really trace back a specific microbial species that are responsible for the production of metabolites that are implicated in disease pathogenesis. By using these gems, we are additionally able to simulate different diets. As a pilot study, we simulated two different diets for one case subject who developed celiac disease at the age of 18 months, where the subject is on gluten-containing solid foods simulated by a typical Western diet, and then compared it with the microbiota of the same subject at 4 to 6 months of age, where the subject is on the breast milk diet. Again, we were able to not only determine the metabolites that are differentially produced between the two diets, but we were also able to trace back microbial species that are responsible for the production of these metabolites under the breast milk diet or under the solid food diet or under both diets. To summarize, the important takeaway from this study is that Gems of Metabolism enable fine-grained functional profiling of the microbiomes. And this is because these models can encapsulate information about which species carry the necessary pathways or transport mechanisms needed for the production and secretion of specific metabolites. And when combined with dietary data, these models can be used to predict species growth and metabolite exchanges among microbes, from which we can tease out which microbes are responsible for the production of metabolites that might be implicated in disease pathogenesis. Thanks for listening, and please feel free to reach out to me with any questions that you may have.